A big, warm welcome to today's CELLIMA Talks webinar, Guardians of Health, the study of ion channels in early drug discovery. Joining us today is Thomas Müller, a distinguished scientist with over 30 years of experience in ion channel research. From his early days studying biology in Heidelberg and Glasgow to leading cutting-edge high-throughput screening at Bayer, Thomas has been at the forefront of scientific innovation. In this insightful session, Thomas will share his remarkable journey and delve into the fascinating world of ion channels. Discover how these tiny structures play a crucial role in everything from muscle contraction to neural activity, and how they have become key targets in drug development. Thomas will also highlight groundbreaking technologies such as electrophysiology, optogenetics, and other high-throughput screening methods that have transformed the field. We wish you a pleasant, exciting, and interesting webinar with the Guardians of Health. Enjoy it.
Hello, Thomas. It's great that you have time to join us today for this webinar on the topic of ion channels. Thanks again for joining us. Could you take a moment to introduce yourself to our audience? Let them know a bit about you, what you do, where you're from, and what you're interested in.
Ja, erstmal vielen Dank, Stefan, dass du auch an mich gedacht hast oder dass du mich eingeladen hast.
Sehr gerne.
Und über Ionenkanäle zu sprechen, es macht mir immer Spaß, ja.
First of all, thank you, Stefan, for thinking of me and inviting me. I always enjoy talking about ion channels. My name is Thomas Müller. I am originally from Heidelberg, studied biology in Heidelberg and Glasgow, did my PhD in Heidelberg, at the time when Bert Sackmann and Erwin Neher won the Nobel Prize in Heidelberg for the development of patch clamp electrophysiology.
Also klassische Elektrophysiologie, bin dann zu Bayer gewechselt, wo ich dann die nächsten 30 Jahre
I worked there on glial cells and did classical electrophysiology, then moved to Bayer, where I have been for the last 30 years in various positions. For the last 10 years, I have been head of cellular high-throughput screening here at Bayer in Wuppertal.
Zelluläres Hochdurchsatzscreening hier bei Bayer in Wuppertal. Over
the years, as a project team member, I have brought about 5 or 6 new chemical entities to the clinic. Always in early research, so the red thread through all the things that I have done have always been the ion channels and the various new technologies that have been involved over the years. Things that I was involved in as an alpha or beta tester, such as voltage-sensitive dyes, automated electrophysiology, optogenetics for screening, robo-scissors, multi-electrode arrays. I could go on and on. I think it comes across a little bit. I am a passionate scientist. That is what drives me. And I have been working continuously as a channel worker for 30 years.
Seit 30 Jahren kontinuierlicher Kanalarbeiter.
Das Kanalarbeiter klingt gut. Ich finde es auch klasse.
The channel worker sounds good. I also think it's great how it comes across, especially for people who don't have much experience with research and laboratory work. There was a lot of technology in your story, including a lot of automation, miniaturization, robots, and a lot of programming. I think that image of the researcher as a lone warrior simply no longer exists. It makes no sense nowadays. And even someone who does something like Mr. Fleming in his laboratory with one hand, comes back the next day and accidentally finds the earth-shattering result, that's not how research is done anymore.
Ich kenne keinen mehr.
Definitely.
Let's move on to the first question. Today's topic is ion channels, one of your pet subjects. We have named the title, Guardians of Health, the study of ion channels in early drug research. Can you explain a little bit? What are ion channels actually? What are their basic functions? When do we actually notice in everyday life that something is not working properly with regard to our health? What are the medical aspects behind it?
Ja, also erstmal, seitdem ich letztes Jahr gelernt habe, dass selbst Viren Ionenkanäle haben, glaube ich, es gibt keine Zelle,
Yes, since I learned last year that even viruses have ion channels, I don't think there is a cell that doesn't have an ion channel. And so even bacteria already have ion channels, and probably the original function of ion channels. Or, in other words, ion channels are pores through which ions flow, as you would classically imagine in a channel.
In einem normalen Kanal fließt aber da ziemlich alles durch, je nachdem, wie groß dieses Loch ist.
But in a normal channel, pretty much everything flows through, depending on how big the hole is. The ion channel is characterized by the fact that, depending on the structure, i.e. in the molecular, biological, three-dimensional structure of the protein, how the charge is in the pore, and depending on this, certain ions can flow through. What the channel does not do is regulate the direction in which the ions flow. In other words, the direction in which the ions flow depends on the ion gradient between the outside and inside, but the channel decides whether chloride ions flow through, whether sodium ions flow through, etc., etc.
Kanal haben sich die Funktionen der Kanäle so weiterentwickelt, dass es eben wegging von diesen reinen Detektieren.
Over millions of years, the function of the channels has developed in such a way that it has moved away from these rows that I have in the environment to a totally diverse function, whether that's muscles, whether that's thinking, whether that's hearing. You could probably go on. And in principle, there are two main types of channels. One is the voltage-activated channels.
Die sind im Prinzip dafür zuständig, wenn man so ein bisschen schwarz-weiß malt, dafür schneller.
In principle, these are responsible for triggering rapid activity if you paint it in black and white. When I want to transmit an excitation signal directly to the muscle, when I gasp for air, so to speak, in the heart, very centrally.
Im Herzen ganz zentral. Es gibt keine Gehirnfunktion ohne Ionenkanäle, kein Denken.
There is no brain function without ion channels, no thinking. On the other hand, there are these so-called ligand-activated channels that somehow determine these gray areas, i.e. not yes, no, but decide in between, whether it's about emotions, whether it's about memory, about fine-tuning ourselves. And you can easily imagine that whenever there is a malfunction because a channel is not working properly, because there is perhaps too much of a ligand that activates the channel,
Weil eine Punktmutation da ist, weil es Substanzen gibt.
because there is a point mutation, because there are substances that bind to the channel that should not actually be there, for example, drugs such as ecstasy or similar, then, of course, this can lead to massive side effects, and many of the diseases, especially with the heart or with the brain, have to do with the channels, and that was precisely one of the reasons why I was actually particularly interested in neurobiology back then as a student.
Ja, klar. Du hast gesagt, Ionenkanäle gibt's, also gibt oder so rum. Es gibt keine Zelle, die keine Ionen.
You just said ion channels exist everywhere. Or, in other words, there is no cell that does not have ion channels. So they are very important. You also hinted that they are particularly interesting and important for medical aspects, especially for human health. Which ion channels are currently being researched for medical treatment, and why are they the ones that have been selected for this, and which are perhaps considered to be particularly promising?
Okay, also zum einen, es gibt schon eine ganze Reihe bekannter Medikamente, die an Ionenkanälen wirken.
Okay. Firstly, there is a whole range of known compounds that act on ion channels. Classic cases are the sulforyl ureas, which act on the potassium channels of the pancreas, the cells there, and thus help with diabetes, so to speak. They were at least known in the, I think, 50s, 60s, 70s, and were essentially developed back then. They were the drugs of choice for treating diabetes for a long time.
Wirklich richtig coole neue Substanzen entwickelt worden sind.
New things are cystic fibrosis, where really great new substances have been developed. And Bayer, of course, where I work, has a history with the so-called dihydropyridines, which were developed for blood pressure treatments, where Adalat also came out as part of Bayer.
Ich glaube, wo immer Krankheiten da sind, die noch einer guten Behandlung ausharren,
Nowadays, I believe that whenever there are diseases that are looking for a good treatment and ion channels are involved somewhere, pharmaceutical research is very happy to work on them because ion channels are considered to be proteins that can actually be addressed well with chemistry, with small molecules.
Darunter fällt Vorhofflimmern, das Herz, ganz klar.
This includes atrial fibrillation, the heart, of course. This includes pain, which brings us back to the brain, or peripheral pain transmission. In recent years, we have been working on sleep apnea, where an ion channel has been linked to it.
Auch da, ich glaube, das könnte man relativ weit weiterführen.
I believe that this could be taken relatively further,
Einfach weil man weiß, man hat eine gute Chance.
simply because we know that we have a good chance of finding something in order to then test in a disease model, so to speak, whether this working hypothesis that this ion channel actually plays a role in this disease is correct, and whether we can then offer patients a new drug in the end.
Jetzt arbeitest du nicht beim, sondern im Hochdurchsatzscreening, im zellbasierten.
You work in cell-based high-throughput screening, and that's where you can test millions, at least that's what you do, millions of potential active ingredients at the same time to see which substances are the most promising, which ones you might be able to develop into a new drug. Can the screening technology also be used to search for new therapies for ion channels, or has that already been done? And how has screening changed the search for ion channels?
Ein Game Changer. Und zwar ursprünglich als Elektrophysiologie, ich hatte vorhin ja schon gesagt, mal noch näher erzählt, was die Elektrophysiologie oder was die Ionenkanalforschung so speziell macht, aufgrund dieser...
So absolutely. In my opinion, that was a game changer. I already mentioned earlier what makes electrophysiology or ion channel research so special, where you can go onto a cell with a glass pipette. I actually have the unique opportunity for biology to see in real time, in reality, how such a channel opens and closes. It opens and closes. And I can follow this directly online. Totally great, totally fascinating, and extremely impressive.
Diese Messung kann man sich ja vorstellen, wenn ich so ein kleines Stück...
The problem is, you can imagine this measurement when I have such a small piece of membrane or pipette attached to the front. It's very delicate. It doesn't last long. It's really exhausting for the scientist sitting in front of it. So you can sit there for a week and prepare the measurement before you can actually measure it. So that substances, we now have a substance library at Bayer, which is not much different at other large companies, of a few million substances that is not feasible. So electrophysiology is a great method, but not for finding initial structures or new substances.
Es ist vor etwa 20 Jahren ist uns neue Farbstoffe gefunden worden.
Now, about 20 years ago, new dyes were discovered that were originally developed for photography. These are voltage-sensitive dyes that spread passively across a membrane. They are charged, and they can be used to derive the membrane potential in cells so that it was suddenly possible to test a huge number of substances very quickly on any channel using such a dye in a microtiter plate, which you have to imagine is a small plate with 1,500 holes in it.
Total schnell eine riesige Anzahl an Substanzen an einem beliebigen Kanal zu testen. Also ein bisschen Gefühl zu geben. Wir waren dadurch dann in der Lage, So
to give you an idea, we were able to test 1 million substances in a week. Of course, this gives us a completely different variety, a completely different chemical space, which enables me to find things that are not yet known, and also to find out quickly whether I have found something that is specific.
Was auch spezifisch ist. Wie potent ist die Substanz?
How potent is the substance? Does it do something to other channels, other proteins?
Es ist uns, aber auch ganz vielen anderen Pharmafirmen wirklich gelungen.
And this has also enabled us and many other pharmaceutical companies to find completely new structures, new tools with which we have been able to develop new drugs or to make substances available to scientists at the academy in order to test ion channel effects in a physiological context.
Im physiologischen Kontext zu überprüfen.
Mhm. Bei den Farbstoffen. Du hast Farbstoffe angesprochen, die weiter den großen Meilenstein gebracht haben bei der Messung von diesen Millionen von Substanzen. Fällt mir als
Regarding dyes, you mentioned dyes, which have brought another major milestone in the measurement of these millions of substances. As a fluorescence biologist, I call myself a bit like that, I also think of bioluminescence. So, the glowing animals, the jellyfish, the glowworms, whatever you can imagine. They are not only found in nature, but are also used for scientific experiments, not the animals, but the fluorescent proteins. And in recent years, many innovative new measurement methods have been developed and established that are already being used quite well and efficiently. It is also said that these new methods are particularly sensitive. Do these self-luminous fluorescent proteins also help to better understand the concepts and functions of ion channels?
Jein. Also ja, sie helfen, sofern man die richtigen Fragen adressiert. Oder andersrum wieder gesagt, ich hatte ja vorhin gerade gesagt, Elektrophysiologie, Patchclimb, ich kann am Molekül direkt sehen, was passiert.
Yes, they help, as long as you address the right questions. And to put it the other way around, I mentioned electrophysiology and patch clamp earlier. I can see directly from the molecule what is happening. On the other hand, the discovery of the dye that detects calcium, the further development of fluo, has of course made it much easier to observe calcium channels in tissue or cells.
Unheimlich vereinfacht. Und dann sind wir wiederum auch beim Durchsatz.
Then again, we have the throughput. The use of voltage-sensitive dyes, which did not come from nature but from photography, suddenly helped us to do high-throughput screening.
Es gibt aber auch ganz andere, neuere Sachen, also neuerst in dem Fall, ich glaube so etwa 10 Jahre.
But there are also completely different, newer things, the latest, in this case I think about 10 years old, optogenetics. This in turn comes from, or optogenetics in this case, means that there is a specific ion channel in our eye that reacts to light, rhodopsin, and which is then activated by light, so to speak, opens and lets ions through.
Dass man zum Beispiel
And this has been adapted to such an extent that such channels have been artificially expressed in tissue, for example. And using this, it was then suddenly possible to activate the channels via light and, for example, to change the potential of cells for milliseconds and thus specifically observe the function of channels in a specific tissue and only where the channel occurs and where it has been activated by light.
Der Kanal vorkommt und wo man ihn durch Licht aktiviert hat.
We also use this for screening because otherwise we would have had to pour substances back into our small holes in the microtiter plate, which we would never be able to get out again, but we were then able to clamp the cell for a short time using the short light pulse and then observe the function of voltage-activated channels, for example. On the other hand, these dyes, these bioluminescent dyes such as Fura, etc., are generally very slow. Electrophysiology, however, allows me to observe a very fast channel. So I think the point is, it's good if our Swiss Army knife of tools that we have to study ion channels, if that increases, but there is not a one-fits-all technology here.
Also Technologie, sondern die Fragestellung ist das Wichtige und wie ist.
But the question is the important thing, and which technology is the most suitable to answer exactly that question.
Und diese Technologien tauschen, also die
And these technologies do not cancel each other out, but rather complement each other to be able to map the overall picture.
Perfekt, genau so ist es. Sie ergänzen sich und am Ende ergibt sich dann quasi.
Absolutely perfect. That's exactly how it is.
Einen Farbstoff, also bei uns ist es.
That complements each other. So for us, the classic approach is to do a screen, test a few million substances, find substances that show that membrane potential changes occur. Then we go to electrophysiology and see whether this is really the case. Then we verify this. We are then interested in how this effect, which we have seen in the isolated channel, is transmitted in a cellular context, ideally in a tissue. The dyes then play a very important role again.
Absolut, genau.
Ja, trotz dieser neuen Methoden, Biolumineszenz, trotz Hochdurchsatzscreening, ist es ja nicht ganz so einfach.
Despite these new methods of bioluminescence, despite high-throughput screening, it is not so easy to find new drugs. In particular, it is difficult to find ion channels that have a particularly high potential and then to identify and capture them for new therapeutic purposes. In your opinion, what are the biggest hurdles that scientists face in the development of drug candidates for ion channels?
Hängt wieder vom Target ab. Ja? Und damit wieder von der Frage.
This again depends on the target, and therefore on the question. Let me illustrate this. So two big things are certainly specificity, which means ensuring that a substance only ever works in one channel, and the other is side effects. Let me briefly illustrate what I mean by specificity. As I said earlier, the potassium channel already exists in bacteria. The pore of the potassium channel, where everything has to pass through, is almost identical in a bacterium to that in us, in most potassium channels. Now, you can easily imagine that if you have a pore like this and are looking for substances that somehow inhibit this channel, then most substances go into this pore. So really, in the classic way, they clog the pore.
Glaube ich, ganz einfach sich vorzustellen, wenn diese Pore jetzt von ihrer Form her bei allen Kaliumkanälen gleich ist.
I think it's quite easy to imagine that if the shape of this pore is the same for all potassium channels, then of course the probability is that if I find a substance that enters this pore, it will be difficult to do this selectively. So for example, when we screen, that's always a catchphrase, finding an antagonist, a blocker for a potassium channel is very easy. Finding a specific one is almost impossible.
Man irgendwie versucht, sein Essay so aufzubauen, dass man eben ganz bewusst.
There is, of course, a strategy of somehow trying to set up your assay in such a way that you deliberately find substances that do not enter this pore or domains of this channel, accessory subunits, etc., that do not occur in the others, because this makes it easier to find selectivity. Or if this is not the case, via very rigorous testing, even in high throughput, of structurally related channels, because if you can achieve specificity, there is a high probability that this specificity will actually apply to other channels. So I think this can be addressed, but it is often difficult enough. There are quite a number of highly attractive potassium channels, especially for atrial fibrillation, where we have so far failed because it is selective, because we cannot find selective binding.
Weil eben ein Kanal leider eben nicht nur.
The other problem is the side effects. Unfortunately, a channel is not only expressed in the atrium of the heart, but also in the brain, in other areas such as the lungs and so on.
Kanal zu Kanal unterschiedlich, wo man eben nicht möchte, dass dort eine Wirkung auftritt.
Of course, you don't want it to have an effect there. For example, we once worked on the potassium channel. The substance did exactly what we wanted it to do.
Hochspezifisch, hochselektiv, potent.
It was highly specific, highly selective, potent agent against atrial fibrillation. But if we want to address the atrium, the substance has to get into the blood. And it does this in a whole range of other tissues. And there it doesn't work in the way we porpoised. That's why these substances sometimes don't even make it into animal experiments and certainly not into the clinic.
Womit, glaube ich, oder wo eine Strategie für Ionenkanal Drug Hunter.
I believe that one strategy for ion channel drug hunters and pharmaceutical companies is as follows. How can we find substances that only work very selectively on the very specific area? For example, can we manage to transport or express a substance in such a way that it only occurs in the atrium?
Können wir es schaffen, dass wir Substanzen finden, die einen gewissen Zustand, also ionenklassischer Spannungsaktivität.
Can we manage to find substances that have a certain state, let's say a classic voltage state of the ion channels? There are certainly a number of other states. Can we manage to address a very specific state? The aim is to address this specific state of such a channel. If the same state is also present in other tissues, but the channel is not active there, we want to achieve functional selectivity. But this is definitely one of the biggest challenges.
Ist aber mit Sicherheit eine der großen Herausforderungen.
Mhm. Strategien ist das Stichwort, das du gebracht hast. Und wie kann man dann Medikamente...
Strategies. It's the key word. How can we find drugs that influence ion channels in unexpected, unconventional places? And how can we use this to enable new therapeutic concepts?
Also ich glaube, bin mir nicht ganz sicher, ob man das sowas als neues Therapiekonzept bezeichnen würde, weil der Mechanismus ist lange bekannt, aber der mit Sicherheit gerade für die ligandenaktivierten Kanäle, also die
So, I don't think I'm quite sure whether you can call this a new therapeutic concept because the mechanism has been known for a long time, but the mechanism is certainly the same for ligand-activated channels, which need a substance and then the pore opens, and in contrast to the voltage-activated channels, which need the voltage, this is allostery.
binden und machen nichts.
And there are so-called positive allosteric modulators, which, for the sake of simplicity, bind these substances to the binding pocket, which initially has nothing to do with the ligand binding pocket. They bind and do nothing. But what they do is change the conformation of the channel so that the ligand can bind better, resulting in a shift in activity.
Allosterische Modulatoren, die den GABA-Rezeptor aktivieren.
For example, substances that some of the listeners may be familiar with are Valium, Librium, these classic sleeping pills, which are partly positive allosteric modulators that activate the GABA receptor, which hyperpolarizes the cell, and this makes you calmer. In other words, what these substances do is that they can only then, they are there all the time and do nothing until the channel is activated, and then the channel can have a stronger effect because there is less ligand. So a classical ligand, an artificial, classical ligand would be in the channel all the time, so you can easily imagine that side effects are much more easily possible.
Auch da wiederum, Entschuldigung, wenn ich ins Wort falle, auch da gilt wiederum, zentral ist zu fragen, was suche ich?
Here too, the central question is, what am I looking for? Am I looking for a substance because, for example, a patient has much less of a channel? Then I would prefer to activate this channel all the time.
Die Aktivität von einem Kanal verstärkt.
Or am I looking for something that only increases the activity of a channel at a certain time? Then I would take an allosteric. Or am I looking for something that expresses a channel less or expresses it more? This is where all drug research begins. It starts with this central question. And then, what am I looking for? And the next step is then logically, what does the ideal biological test system look like to find something like this?
Ist es denn hilfreich, wenn diese allosterischen Zentren kovalent gebunden werden?
Would it be helpful if these allosteric centers were covalently bound?
Jein.
Ja.
Ja, kann natürlich sein. Das Problem, was man normalerweise mit den Kovalenten hat, also bindet dauerhaft,
Genau, ja.
das ist eine dauerhafte Wirkung.
Yes, of course, that can be the case. The problem you normally have with covalent, permanently binding substances is that you then have a permanent effect. That's usually what you want. However, there is a greater risk of side effects.
Was es gar nicht so häufig gibt.
Okay.
Das andere
I have to be honest, Stefan, I'm not so sure about that. I think that is something that is not so common with ion channels. The other thing, which is, of course, the charm, is that with many other proteins that have a very long half-life, some of which are there for weeks, if I then have a covalent binder, it will inactivate this protein for the entire time that it is there. However, ion channels are often only in the membrane for a relatively short time. So even if I have a covalent binder, if the channel is transferred into the cell after a day and is degraded there, it will be gone again relatively quickly. So although you have a covalent binder, the effect of the substance is then relatively manageable.
Spannend. Jetzt haben wir schon
Fascinating. We have talked about the heart, the brain, these two prominent organs. And then we also talked about the fact that ion channels are actually found in all cells. And now there is this disease in an aging society, which is called cancer. What role do ion channels actually play in cancer therapy? If they are found everywhere in all cells, it is obvious that they can be addressed. Can they be used in a targeted manner to treat certain types of tumors?
Interessante Frage. Also ich habe ja vorhin schon erzählt, ich komme eigentlich aus der Neurobiologie her. Dabei habe ich sehr viel, also vorübergehend der Diabetesforschung und dann eben sehr viel in der Herz-Kreislauf-Forschung gearbeitet.
Interesting question. As I said earlier, I have a background in neurobiology. At Bayer, I worked a lot temporarily in diabetes research and then a lot in cardiovascular research.
Kanal, TAS-Kanäle, KV-Kanäle. Einige von denen sind sogar Biomarker für Krebs.
Yes, potassium channels in particular have long been associated with cancer cells. There are a number of ion channels, for example, the ERG channel, the ether-a-go-go channel, TASC channels, potassium channels. Some of them are even biomarkers for cancer. Interestingly, I'm not aware of really many pharmaceutical companies working on ion channels to treat cancer cells. I'm also not sure whether the ion channels are causal or a reaction of the cancer cell to the increased growth, to the altered oxygen content in the tissue, to which the cells then react with an increased expression of the channels. Off the top of my head, Stefan, really off the top of my head, I've never thought about it before. Perhaps it would be an interesting concept to address non-potassium channels as a cancer target, for example, in order to treat cancer. Maybe it would also be a concept, together with a classical anti-cancer drug and a potassium channel antagonist, to enhance the effect of these classical anti-cancer drugs, so to speak. I'm sure with the thousands of cancer researchers out there, someone has looked at this. I'm not an expert, but it's really interesting that compared to many other diseases, ion channels don't actually play a prominent role in cancer research.
Ja, würde denn immer Computer werden eingesetzt, Roboter werden eingesetzt.
Computers are used. Robots are used. Then you use big data. I'm thinking of scientific big data with data mining. Now, there are deep learning and machine learning approaches and models that are used to improve our understanding of ion channels. Can these approaches also be used for this purpose? And are such models important at all? Or do they even provide a certain degree of prediction when it comes to finding new drugs?
The first answer would be that ion channels are actually predestined for this, because there was also a Nobel Prize for this. And there are models from the 1940s and 1950s, very simple models that can be used to calculate the membrane potential. So these are the classic ones. Anyone who has studied biology or any biology student will remember the Goldman equation, Nern's equation, Hodgkin-Huxley, which can really be used to model an action potential.
Ich weiß noch, dass als ich in Heidelberg war, und damals die Laptops noch Schlepptops hießen, weil man sie
I remember that when I was in Heidelberg and the laptops were still called drag tops back then because you had to carry them in pairs. We had developed a program, for example. It was one of the first computer programs for teaching at university where you could really model everything. You could model action potentials. How do toxins affect action potentials? How do action potentials reach the all-or-nothing threshold?
Man kann basierend auf der Goldman-Gleichung, wenn ich weiß, welche Kanäle in meiner Zelle vorkommen, kann ich direkt sagen, wie das Membran.
Based on the Goldman equation, if I know which channels are present in my cell, I can directly say what the membrane potential of the cell is. Conversely, I can specify exactly how many ion channels there are. Actually, you could say that it is predestined for the use of artificial intelligence. The problem is that we usually don't know exactly which channels are present. To make the whole thing even more complicated, they are not only located in the membrane, but some of them are also located in vesicles. Sometimes there are also these so-called microdomains, where, for example, the third one, where the ion ratios are completely different locally, so that the channel, although it is exactly the same channel, suddenly has a completely different effect due to the different ion gradients.
Was für eine Rolle ein Ionenkanal in einem menschlichen Gewebe spielt mit künstlicher Intelligenz unheimlich schwer ist.
So predicting what role an ion channel really plays in human tissue is incredibly difficult with artificial intelligence. To illustrate, heart attack. You would think that a heart attack would happen or a cardiac arrest would happen if you somehow block the large arteries. It is often a very small artery somewhere that can suddenly lead to a heart attack.
Sodass, ja, es gibt mittlerweile sehr gute Modelle, mit denen man auch so Weiterleitung von Erregungen im Herzen modellieren kann und damit gewisse Vorhersagen treffen kann.
Yes, there are now good models that can be used to model the conduction of an excitation in the heart and make certain predictions. But a detailed or accurate prediction is not possible, especially for the heart. On the other hand, we need to ask our colleagues in safety pharmacology about this, because there are certain channels where many drugs that have nothing to do with ion channels still work.
Es stimmt jetzt nicht ganz, was ich sage, aber es veranschaulicht so ein bisschen dieses Verstopfen von dem Kanal, kann man sich leicht vorstellen.
It's not quite true what I'm saying, but it illustrates this clogging of the channel. It's easy to imagine. This leads to cardiac arrhythmia. LQT syndrome is known for this, so certain ion channels actually have to be tested before a substance can be administered to humans.
Meines Wissens nach gibt es tatsächlich schon eine ganze Reihe von Programmen, mit denen man Dinge simulieren kann.
And as far as I know, there are already a whole series of programs that can be used to simulate things. But because the channel, the structure, where they occur, what role they all play is very well known,
Zu vereinfachen.
These models are now being used to simplify predictions so that we don't have to enter these models with every substance.
Wo man so ein bisschen die Vorstellung hat, naja, also.
So, yes, it helps. But if you have a bit of an idea based on this Goldman or Hodgkin-Huxley equation, it should actually be quite simple. No, nature is much more complicated than that.
Was könnte man denn machen, damit man diese Modellierung und das ganze KI, Deep Learning, Machine Learning, Werkzeug, das ganze Sammelsurium verbessern kann, damit die Simulationen auch besser sind?
What could be done to improve these models? The entire AI, deep learning, machine learning tools, this whole conglomeration, so that the simulations also improve, so that they have less bias due to the information with which they are fed, so that you can really get a more differentiated picture of ion channels, how they work, how they interact with drugs, and that these computer-aided systems not only have innovation potential, but also actually make a decisive contribution to drug research.
Damit kein falscher Eindruck entsteht, das tut sie schon.
Okay.
Also, indem man im Prinzip, also wenn ich es gibt eben sehr gute.
To avoid giving the wrong impression, it already does. By basically, there are already very good models in various tissues of how excitation is transmitted, for example. And I can use models to make predictions about what my substance should do there.
Darüber kann ich dann, wenn das dann so ist, kann ich mir Gedanken machen.
If this is the case, I can then think about what an experiment looks like with which what I have just predicted here in silico, so to speak, fits.
Und kann darüber dann wiederum meine Substanzen vielleicht auch verbessern.
And can then perhaps improve my substance. Structural biology. Stefan, we know that there is a whole range of proteins where very good predictions are now possible.
Mittlerweile, also bei Ionenkanälen ist es ein bisschen schwieriger, weil es eben diese verschiedenen Zustände gibt.
In the meantime, it seems it's a bit more difficult with ion channels because they have different states. I am sure that we will make so much progress in the next 15 years that it will be possible to better predict where binding pockets might be, and perhaps also to develop substances for them.
Umgekehrt hatte ich vor kurzem auch gehört, dass es tatsächlich auch möglich ist, über AI künstliche Proteine herzustellen.
Conversely, I recently heard that it is actually possible to produce artificial proteins using AI, which in turn bind to natural proteins and change their effect.
Ganz trivial. Also wenn ich hier alle paar
There are so many different areas of application. Screening, quite trivially, if I screen 5 to 10 targets on 5 million substances here every year,
zu überprüfen, welche Substanzen sind selektiv, wie sind die an anderen Substanzen.
of course, artificial intelligence helps me to check directly which substances are selective. How have they been found in other substances on other targets? How do I have to optimize this? So I think there are many areas in ion channel research where artificial intelligence is already helping us. However, it is certainly not the case that we have the sole solution and that it will help us with every problem.
Das ist ein gutes Schlusswort auch.
That's a good closing statement, too. AI or the new AI methods at the moment, they are important, but they are not the be-all and end-all.
Ich hatte, erst kürzlich war ich auf einer Tagung, hatten wir einen Workshop hier, wo mir
I was at a conference recently, and we had a workshop where a professor from Cambridge spoke to me from the heart about machine learning.
Disease understanding.
Disease understanding. We need to understand the disease. If we want to understand the disease, if we want to know where we have gaps, we have to ask the right questions.
Für diese Fragen müssen wir die richtigen Testsysteme entwickeln.
For these questions, we need to develop the right test systems. We need to generate as much data as possible for these test systems.
Dann hilft uns AI.
This data needs to be as good as possible. Then AI will help us move quickly from these experiments to the clinic and to a successful drug.
Mit schlechten Daten hilft uns.
With bad data and a wrong experiment, it's trivial. No artificial intelligence will help us to find a new substance.
Eine wahre Aussage. Thomas, ganz herzlich.
A brilliant statement. Thank you very much for the fascinating conversation and the detailed insights into ion channel research.
Faszinierend.
Fascinating for me as a simple cell biologist.
Ja.
Thank you, Stefan. See you then.
Klasse. Danke.
Thank you.
Thank you very much for your participation, time, and attention. We look forward to see you again for an exciting and informative science talk. Until then, stay well and take care.